Welcome to Module 1 of our course on the metabolism of lipids. In this session, we'll start by building a strong foundational understanding of lipids, what they are, how they are classified, their chemical and physical properties, and their biological significance. From there, we will move into how lipids are digested, absorbed, stored, and mobilized, along with the important disorders linked to lipid metabolism. Finally, we will explore the oxidation of fatty acids, a critical energy-yielding process. 1. Introduction to lipids. Lipids are a diverse group of hydrophobic or amphipathic molecules found in living organisms. Unlike proteins and carbohydrates, lipids are not defined by a specific repeating unit such as amino acids or monosaccharides. Instead, they are defined by their physical property of being insoluble in water but soluble in nonpolar organic solvents such as ether, chloroform, and benzene. Functionally, lipids play multiple roles in biology. Energy storage. Lipids provide more than twice the energy per gram compared to carbohydrates or proteins. Triacylglycerols are the main storage form. Structural components. Lipids form the structural matrix of biological membranes, such as phospholipid bilayers. Signaling molecules. Certain lipids act as hormones, second messengers, or regulators of cellular activity. Insulation and protection. Stored fat cushions organs and provides thermal insulation. 2. Classification of lipids. We classify lipids into several broad categories based on their structure and function. 2.1. Fatty acids. Fatty acids are the simplest lipids, long hydrocarbon chains with a terminal carboxylic acid group. They may be saturated, contain no double bonds, typically solid at room temperature, e.g., palmitic acid. Unsaturated, contain one or more double bonds, often liquid at room temperature, e.g., oleic acid. Polyunsaturated fatty acids, PUFAs contain multiple double bonds, such as linoleic and linolenic acids, which are essential for humans. The chain length and degree of unsaturation influence melting point, solubility, and biological function. 2.2. Triacylglycerols, triglycerides. These are esters of glycerol with 3 fatty acid chains. They are the main storage form of energy in adipose tissue and are highly efficient for long-term energy storage because they are anhydrous and compact. 2.3. Glycosylglycerols. Lipids that have one or more sugar residues attached to glycerol. They play important roles in cell recognition and membrane stability. 2.4. Phospholipids. A major component of cell membranes, phospholipids have a glycerol backbone bound to 2 fatty acids and a phosphate group, which is often linked to another molecule such as choline or ethanolamine. 2.5. Sphingolipids. These lipids are based on sphingosine rather than glycerol. Examples include sphingomyelin, which is important in the myelin sheath of nerve cells. 2.6. Waxes. Esters of long-chain fatty acids with long-chain alcohols. They are protective coatings in plants and animals. 2.7. Prostaglandins and related eicosanoids. Derived from arachidonic acid, prostaglandins, thromboxanes, and leukotrienes are important signaling molecules involved in inflammation, vascular tone, and other processes. 3. Physical organization, micelles, monolayers, and bilayers. Lipids in aqueous environments can self-organize due to hydrophobic interactions. Micelles, spherical structures with hydrophobic tails inward and hydrophilic heads outward. Monolayers, a single layer of lipids, often seen at the air-water interface. Bilayers, 2 layers of phospholipids with hydrophobic tails facing inward, forming the basis of cell membranes. These structural arrangements are critical for understanding lipid digestion, absorption, and transport. 4. Digestion of lipids. Lipids present a challenge for digestion because of their hydrophobic nature. 4.1. Emulsification. Dietary lipids are mostly triacylglycerols, which are not soluble in water. In the small intestine, bile salts, derived from cholesterol in the liver, emulsify fat droplets, breaking them into smaller micelles and increasing surface area for enzymatic action. 4.2. Enzymatic hydrolysis. Lingual and gastric lipases begin fat digestion in the mouth and stomach, especially important in infants for milk fat digestion. Pancreatic lipase, the main enzyme responsible for breaking triacylglycerols into 2 monoacylglycerols and free fatty acids. Phospholipase A2, cleaves phospholipids. Cholesterol esterase hydrolyzes cholesterol esters. 5. Absorption of lipids. Product of digestion, monoglycerides, fatty acids, lysophospholipids, and cholesterol are incorporated into mixed micelles with biosalts. These micelles transport lipids to the brush border of intestinal epithelial cells. Inside the enterocytes, lipids are reesterified into triglycerols, phospholipids, and cholesterol esters. These lipids are packaged with proteins into chylomicrons, which enter the lymphatic system and then the bloodstream. 6. Storage of fats. In adipose tissue, triglycerols are stored in large lipid droplets. The adipocyte serves as the body's energy reservoir, releasing fatty acids during fasting through lipolysis, a process mediated by hormone-sensitive lipase activated by catecholamines. 7. Disorders related to lipid metabolism. Several inherited or acquired disorders affect lipid metabolism. 7.1. Obesity. Excessive fat accumulation due to imbalance between energy intake and expenditure. 7.2. Carnitine deficiency. Carnitine is essential for transporting long-chain fatty acids into mitochondria for beta oxidation. Deficiency leads to muscle weakness and hypoglycemia. 7.3. Carnitine acetyltransferase deficiency. An enzyme defect impairing fatty acid oxidation, leading to accumulation of fatty acids and hypoglycemia. 7.4. Refsum's disease. A defect in phytanic acid metabolism, leading to accumulation and neurological symptoms. 7.5. Atherosclerosis. Deposition of cholesterol and lipid-rich plaques in arteries, leading to cardiovascular disease. 7.6. Myocardial infarction. Blockage of coronary arteries, often linked to lipid imbalance. 7.7. Lipidosis. A group of inherited disorders of lipid degradation, such as Gaucher's disease, characterized by glucocerebrosidase deficiency. 8. Fatty acid oxidation. Fatty acid oxidation, also known as beta oxidation, is the main catabolic pathway for generating energy from fats. 8.1. Activation of fatty acids. In the cytoplasm, fatty acids are activated to acyl-CoA by acyl-CoA synthetase in an ATP-dependent process. 8.2. Transport into mitochondria. Long-chain acyl-CoAs are transported into mitochondria via the carnitine shuttle. 8.3. The beta oxidation cycle. Each cycle involves 4 steps. Oxidation by acyl-CoA dehydrogenase, producing FADH2. Hydration by enoyl-CoA hydratase. Oxidation by beta-hydroxyacyl-CoA dehydrogenase, producing NADH. Thiolysis by thiolase, releasing acetyl-CoA. Each turn shortens the fatty acid chain by 2 carbons and produces 1 acetyl-CoA, which enters the TCA cycle, generating ATP. 8.4. Energy yield. Complete oxidation of a palmitic acid molecule yields about 106 molecules of ATP.